and following with questions.
Good morning, everyone. My name is Aisling Brenan, and today I'm going to talk to you about microplastics in the North Sea, enhancing our ecological understanding through the development of rigorous sampling methods. My thesis was done in Vlies in Belgium under the supervision of Annelies de Klerk. Everyone knows microplastics and plastics pollution are a huge topic today, but I'm just going to tell you a little bit about what microplastics are and where do they come from. So there's two main sources. You have primary microplastics, which were made to be microscopic in size, and then you also have secondary microplastics, which are from the degradation of macroplastics. Just to give you an estimation of how many plastics might actually be in the ocean, there was reported 348 million tons of plastics produced in 2018, and estimated about 1.4 million tons of microplastics lost to the ocean. That's microplastics alone, so it's quite a lot. Microplastics are separated into three size classes. You have the large microplastics from 5 to 1 millimeters. You have two ranges, the upper and lower range of small microplastics ranging from 999 micrometers all the way down to 1 micrometer. And then you have your nanoplastics in the nanoscale. There's quite a lot of studies at the moment being done on the ecological problems caused by microplastics, such as microplastics involved with physiological stresses, vectors for contaminants such as persistent organic pollutants, and interactions with the marine communities. But what's the point in studying the ecological problems if we can't quantify the microplastics properly? There's been a lot of studies quantifying microplastics, but they always seem to have some sort of constraint, like time, a lower size limit detection, and cost. So at Vlies, they're developing a method for time and cost-efficient method to quantify microplastics down to as small a scale as possible. And they based the method on 6 points. So they looked at cross-contamination reduction, the sampling method, separation of microplastics from the sample, digesting the organic material so you can visualize the microplastics, and then quantifying and characterizing them. So based on those 6 points, I validated the method that's being developed, and I formulated 4 questions. So the first one is, does the digestion method remove organic material without visually affecting the microplastic surface? Is the recovery yield of microplastics from the samples by filtration and digestion method acceptable? Are the sampling and contamination procedures acceptable to apply the methods to environmental samples? And finally, is Nile Red staining combined with fluoroscopy and confirmed by µ-FTIR a fast and reliable method for classifying and therefore quantifying microplastics? For the purpose of the study, I separated microplastics into particles and fibers. So quickly, I'll go through the method that we used. To separate the microplastics from the water, we poured water over a 300 micrometer sieve and onto a polytetrafluoroethylene filter of 10 micrometers to separate the two small microplastic size classes. The sieve and the filter were washed with 33% hydrogen peroxide and left to digest the organic material for 2.5 hours. And after the digestion, they're poured back over new filters, and those filters are stained with Nile Red dissolved in acetone, which has been proven from literature to fluoresce microplastics using fluorescent microscopy. So we developed the fluoroscopy method by viewing stained filters with known plastic particles on them. We looked at them in UV light, blue light, and green light. And we found that blue light was the most consistent for fluorescing microplastics, as you can see here. So they're fluorescent in yellow or orange or white. And so when we were looking at microplastics, we recorded the length, width, the color of the particle or fiber that was found, its position on the filter, and then we took an image of the particle in both UV light and blue light so that we could use it as a reference as to where the particle was on the filter. Because that filter then is moved to a micro Fourier transfer infrared system, where this particle is scanned using infrared light to produce a spectrum. So I scanned a particle, and the spectrum produces the black one, as you can see. And afterwards, that scan is checked with a library database that's on the µ-FTIR, and a percentage match is given for what the particle is the closely related to. And as you can see here, it's a 97% match for polyethylene terephthalate, so it would have said that the particle was plastic. So I'm going to start with the results. So the first question, does the digestion method remove the organic material without visually affecting the microplastic surface? So for this, I looked at the microplastics using a scanning electron microscope. And here's just some examples of nylon, polypropylene, and polystyrene. I looked at them at 300 times magnification and around 1,000 magnification. Untreated are just virgin plastics with nothing done to them. And then the treated is the microplastics after they've been stained with hydrogen peroxide and Nile Red. And as you can see, there's not really any difference between the microplastics. There's no surface degradation, there's no breakdown of particles. The only problem with the digestion method is that when we processed samples from April, they were really algae-laden. So the digestion didn't obviously dissolve them and probably wouldn't, but it would, like a solution that I came up with was maybe a longer digestion time to bleach the algae so they would be less visible when visualizing them. The second question, is the recovery yield of microplastics from the samples by the filtration and digestion method acceptable? So recovery yield is when you add microplastics or anything to the filtration system, how much of them come back to see if it's a representative sample. So we had positive controls, and I added 10 particles of polyethylene terephthalate, around 60 particles of polystyrene 10 micrometer, around 10 particles of polystyrene 20 micrometer, and 10 particles of polyvinyl chloride. I set an upper acceptance limit of 90% recovery, and as you can see, none of the particles actually achieved that, but straight away we found out what the issue was. First of all, at a 90% recovery rate, at particles at such a small size, we needed to add more. But also there was a bit of human error, because here is 110% recovery rate, which means that more particles were added than we expected. And also for here, the count estimation for the polystyrene particles, they were in a solution, so we had to estimate how many beads were actually in the bottle from filtration or from dilutions. So that was quite inaccurate then in the end. And also particles, microplastics have electrostatic behavior, so they probably stuck to the filtration system on the way down. So a solution to that we're working on now is accuracy in counting the particles before they go in and at a higher concentration, and using SDS, sodium dodecyl sulfate, as a surfactant to remove the electrostatic behavior. The third question, are the sampling and contamination procedures acceptable to apply the method to environmental samples? Our sampling seemed to be fine. We made sure that there was no plastics used in, we had a metal bucket. It was stored in glass bottles with metal lid. The only problem is the rope is actually nylon on the Siemensstaven, so we're looking into changing that. And LifeWatch collected our environmental samples from 4 sites off the coast of the Belgian North Sea. We did have a problem with contamination in our negative controls when we started processing environmental samples. And as you can see here, there's 2 images of particles fluorescent in blue light. And it was quite a high count for a negative control. But the second problem was that 51% of these were actually organic and 49% were plastic. So we couldn't tell the difference between organic and plastic particles, which was quite a big problem then. But to help the contamination, we removed the source of organic contamination, which we actually found was the paper that you dry all of the glassware with. So we removed that completely, and we used SDS again between the samples for reduction of cross-contamination, and it brought the negative controls right back down to 0. So the last question is Nile Red staining combined with fluoroscopy and confirmed by µ-FTIR fast and reliable for classifying and quantifying microplastics? So like I said before, we had assumed that blue light, I'm doing this in terms of fibers, just as an example. In blue light, we assumed that they were plastic when they were fluorescent in orange and yellow. And we also assumed that when they were not fluorescent and not visible, they were not plastic. As I said, that did not actually work out. But like I said before, we took images in UV as well, and we started to notice there was a color pattern with UV. So the colors are not really clear here, but this fiber is red and this fiber is pink, and those seem to be correlating to something. We also saw white and blue fibers. And as I was doing µ-FTIR confirmation, I started to find that combinations of blue light fluorescent and UV light red or pink seem to be corresponding to plastics, and any other variation were non-plastic. So I had to test the hypothesis. So I built a decision tree model. I originally had a data set of 41 fibers when I submitted my thesis, but in the last 3 weeks I've built up the data set to 132. And from the things that we recorded, I took the color and I took the µ-FTIR result, whether it was plastic or not plastic. I used 2/3 of the data set to build the model, and then I used 1/3 of the data set to validate it, and I did a 3-fold validation to avoid overfitting in the model, and produced confusion matrices for the reliability of the model. So here's my decision tree. It's really easy to read. If the fiber is blue light positive, UV positive, the probability of it being classified as plastic is 0.92, and the probability of being classified as not plastic is 0.23. Then if the fiber is not blue light positive, UV positive, the probability of it being classified as plastic is 0.03, and then the alternative is 0.9. So from the confusion matrices, I calculated the correctly classified instances as 87% with a standard deviation of 6 and a kappa value of 0.68. And based on literature, I found that a CCI value of above 70% and a kappa value at around 0.6 to 0.8 was substantial. So the model is really reliable, which means the classification of fibers from this method is quite accurate. So to conclude, the digestion method needs to be reviewed so I can visualize the plastics better, but there's no visual effect on the microplastics. The recovery yield is being worked on for the positive controls at the moment, and the recovery yield is increasing. The sampling method is fine, and the contamination method is back down to an acceptably low to no contamination. And indeed, using Nile Red combined with blue light and UV light fluoroscopy is a time and cost efficient manner for quantifying and classifying microplastics. So this implies then that microplastics can be quantified in the North Sea and globally once the method is published, which means that we can collect spatial and temporal data of microplastics concentrations to help with ecological monitoring, and we can also apply the method then to other environments like sediments, like biota, for more concentration of microplastics in the world. So I'd like to thank my supervisor Annalise and the technical supervisor Matthias, everyone in VLIS for helping me out so much, and everyone from Ghent University, and especially everyone from IMBRC for such a great master's.